Finster from Aarhus University, and in Aarhus they have actually built a small little piece of Mars on which they conduct experiments, and will give us some maybe interesting answers about if there's life on Mars now that people do go.
No, I don't think I will give you some answers, but I would like to introduce you to some approaches that we have chosen to study some of the questions which may be interesting. Yeah, myself, I'm an environmental microbiologist that used to work with all different kinds of microbes on Earth. And I used to go out into nature and take the samples, and then I can work with this and isolate organisms and study their physiology. This is quite difficult when you want to do studies on Mars. Until maybe 20 or 25 years, it's not possible to get material from there or to do experiments on Mars. So we have to deal with what we have on Earth. And of course, we're making one big assumption, and then this assumption is that life on Mars is DNA-based life, which looks like the life that we know from Earth, and that it behaves in the same way. And when we accept this assumption, then we can do some studies and ask what would happen if this planet Earth turned into a scenario and went to become Mars. Would life be able to persist under these conditions? And this would be very important when we want to search for life on Mars. As it looks now, it is very almost sure that life is not present on the surface of Mars due to several physical and chemical conditions which are not very favorable to life. But maybe life could persist in deeper strata in Mars. So this would, of course, require digging or at least different kinds of research than the ones that can be done by the rovers that are up there. But I will show you what we we just started with this 2 years ago. So we're in a very early phase of our research, and we came into this by chance. Just the people from the geological department, physics department asked if we were interested in studying some iron minerals in Jutland, which were particular. And we said yes, we could be interested in this, and then from this collaboration, a group started, and we started to develop some equipment which we think could be very useful in these kinds of investigations. Another part would be to look for signatures like Minik is doing. We have the type of signatures we detect them on Mars, then could be a great probability that this has been created by living organisms. Always assuming that they have the same biology, biochemistry, and so forth, like organisms have on Earth. Another thing was we cannot, maybe not looking for organisms, but for molecules which are typically biomolecules, could be proteins, RNA, DNA, which could be survive. How long would these molecules survive under these conditions? And the next question is then, may there be areas on Mars conditions which could improve survival even under these harsh conditions? So we can alter the conditions a little bit. Of course, we could do it and say, okay, but there are areas on Mars that are like Earth, and then we already have proof that life on Earth is possible. So if these conditions prevail somewhere deep in the ground, then there was life in the beginning of Mars, then life, of course, would be there also today. There are three things we could look at. One is can organisms survive, and one of the easy ways for microbiologists just to grow organisms in different media and see if when they still can grow, then they have survived. Another thing is activity. Is there still bacterial activity left after incubations under these conditions? And the next one is diversity. Are there particular organisms which are especially harsh and which can especially suited to survive these conditions? Yesterday we heard a lot about extremophiles, organisms which are adapted to what we call extreme conditions. And they can survive high temperatures, radiation, low temperatures, and aerobic conditions. So we work with the forest soil, and you may not consider forest soil as an extreme environment, but actually it is, because, and even also for bacteria, it's an extreme environment because you have an altering environment here. we consider high temperatures like as an extreme environment, but for most of these organisms, they have adapted to these situations, and actually it's quite stable. If you have high temperatures for many million years in liquid water, then actually the situation is so that you can evolve a strategy to survive under this condition. In forest soil, where you have changing temperatures, you have different conditions of humidity, actually you have a very altering system which actually demands a lot from the organisms. We use this soil also because by our colleagues it is used as mass analog soils in different kinds of experiments. So this was our starting system. And this is our very primitive chamber that we worked with in the beginning. It's just to get started. And you can see here it's just a cylinder which we found in our basement. And we put in a core here, and this core is filled with some soil from the forest area. You can see some cabling here. These are just sensors which allow us to measure the humidity and the temperature in different locations of this soil. When we have regularly, we can measure, we can detect pressure, temperature, we can analyze the atmosphere in the chamber. We have a cooling system, all kinds. And the chamber here is just hidden underneath it here because we don't want to hurt our eyes, so everything is well sheltered for UV radiation. And these were the first experiments. These are our conditions with the temperature. With the lowest temperature here, we also had a measuring point below here. It was about -100 degrees, and in the top it was when we had UV radiation. It was about 12 degrees. There was a large temperature gradient. The pressure is a little bit higher than on Mars. We had UV radiation about 200 nanometer, and this is the intensity. And what we made the first incubation, which was for the UV radiation dose, which is equivalent to 8 Mars days. You can also see from the atmosphere, it's not quite the Martian atmosphere. The oxygen was a little bit higher, and this was due to a small leak in our incubation system. So we constantly had to pump to keep the oxygen pressure low. This was some of our experiments, just to, not that you have to keep these things in mind, it was just to check analysis of the microbial community. These refers to the different chambers that we used. We did an Kultur experiment, which was without UV radiation. Then we had an experiment with UV radiation. 1, 2, 3 refers to the different depths. You can see 0 to 1 centimeter, 2 to 3, and 4 to 5, just to see whether we have effects only in the surface or if these effects reach deeper down into our colors. And what you actually can see is this was our soil. We freeze-dried it, and as you can see, we measure effect already just from the freeze-drying. So in terms of perturability, being in this chamber, being radiated, did not have a tremendous effect on the part of the population that survived and that would be culture apples. We did some utility measurements, and this was down, just looked out of the soil samples and put them in a small container with water on, and then just look for the oxidation of glucose to CO2, because this is one of the important things related to life. Life has always, at least, usually you can detect life by metabolism. When you talk about microorganisms, you also have forms which not metabolize very much like spores, so that would be hard to detect, but usually there's a metabolism. And what you could see here is that in the the upper part, which was the 1 to 1 centimeter, we actually had an effect. It was the only part where we saw an effect. This is expressed as the oxidation rate constant for this glucose. All the others, they are not very much different. You can see that also after some time of incubation, this upper part just came up to the same level as the others. So there was an initial increase in activity in the very upper part of our experiments. Another one we did, it was where we looked for the functional diversity. You see here a plate, and if you look carefully, you can see there are some red dots, and there are some clear. There was a substrate, there was some other stuff. The organism goes, they will salt. There's a setup of 30 substrates in triplicates, and then from the utilization of the cell, you can then see whether this changed as an effect of incubation or position in our tubes. And we have two parts. We have activity, which is just the average color development, and then we have the metabolic diversity, which is the number of substrates, which means the number of different red spots. And there you can see, when you make, for instance, component analysis, that most of the samples are just clustered here, but there was a UV-1 horizon and the UV-2 horizon which actually were different. So these two horizons were affected by the experiment. And the last thing was we used the molecular technique just to look how the molecular diversity was affected by our experiment. Then we used the we used a method where we could separate the predominant species or taxa in our system by a method called penetrating gradient electrophoresis, which is, and all these measurements were based on 16S rDNA, which is the key identification molecule for organisms. In eukaryotes like us, it's 18S, in prokaryotes, it's 16S. And this is on the. And what you see here is a kind of zip code. These are the different lines. And when you compare them, you can use a program which can read these lines and make alignment and then analyze it. We can see that there was hardly any difference. The similarity was more than 88%, which means that there is hardly any dissimilarity. So the diversity was not affected by the incubation that we made. And we were then doing the same incubation, looking into the organisms that could be grown. They can see there is no clear pattern. They all behave. We cannot tell that the number is decreasing. There we have the diversity is decreasing in the samples where you have the highest UV radiation. So the conclusions was here, in survival there was no reduction to incubation, the genetic diversity was not changed, we had a decrease in activity, and the functional diversity expressed in the number of substrates that could be used to utilize the So the factor that induces this change was the UV radiation. All the other factors obviously do not have a large effect on the decrease in diversity. Possible explanations.
Can I just ask, how many generations of bacteria would you have during an experiment?
We have no generations, just 4. They don't proliferate in the system.
Ah, okay.
They are just there.
So there's no genetic diversity, what do you mean then?
No, there was no shift in genetic diversity. We expected that DNA was destroyed.
Ah,
that
okay.
is expressed in a sense. So one of the possible explanations was the incubation was too short. So this is probably true. Then the sampling intervals were not detailed enough, but when we cut it, I told you that we used 0 to 1 and 1 to 2 centimeters, which we actually made 100 microlized them, probably to work with. Then here, another thing is that the DNA is very resistant and is not destroyed. Another thing is the material, so the population was identical in the top and the bottom of our experiment, and this means that the same material in the upper part is in the lower. They are destroyed, not that different from this analysis. And there may be some reactive gases which diffuse into deeper parts, and this may explain the change in activity and also the change in functional diversity. Another thing, and this we also found out in this forest store, spores were mainly present instead of vegetative cell, and these spores are known to be particularly hard, well adapted to these very harsh conditions. Now, here it's not good at here, this is the size of this overhead projector. You can see that it has a chamber. There's a sluice here where we can put in a lock where we could put in samples, and we have checked that all this worked quite well. We can put in 10 samples instead of 1. We have still the possibility to monitor all the other factors that we could monitor before. And the new thing now is that each of these tubes can place a data line, which gives us a very good documentation for the temperature. We also want to be able to measure the UV radiation on top of these tubes so that we can monitor the factors which are important for our experiments. And we hope that by using this piece of equipment, we can run experiments as long as we have months to run them, which means for several weeks or 7 months. And we dream of having an experiment which we could run for a year or so to see if we really have an effect, because I think we have to use these long types of experiments to really be able to tell people about the persistence and the survivability of organisms. Because that's what we can ask. We cannot answer questions about the presence of life on Mars not doing biological experiments. I think there we have to look for what has been talked about before, the atmosphere or other factors which would tell much more about the presence of life. But we can do experiments, reasonable experiments with all these organisms that we have on Earth by incubating them under conditions which reflect the conditions on Mars. And another thing that we do now is that we change our soil. Instead of using the Jutland forest soil, we got permafrost soil. We have permafrost was permanently frozen for many years, and then we assume to have a microbial community which is adapted at these cold conditions. Permafrost not dry, and well, the samples we took are not affected by UV radiation, but they are cold adapted. And other experiments show that here there are many organisms present which don't have spores. So they're probably more susceptible to at least UV radiation and some of the other factors that we treat them with. So what we do now, we go to start with this in this spring, is to do long-time experiments. We have larger sample volumes now because we can work with 10 tubes instead of 1, which means that we can reduce our sampling. It gives us a much better resolution. We can tell under the from joint organisms. Don't say that you can just multiply it by 4 down on Mars. You have to take their organisms, how fast. I always have the possibility to find instrumentation with extend up to masses. That's a very little likelihood to succeed. And then the other thing is that we, by having a data logging system inside our system, it will be possible to work on each individual source. The people are working with this, how very work there. The computers are working with myself and my colleague Ben Lomstein, the biologist, and we have to nurse in physics, and Ben who is the leader.
Preparing for colonization of Mars more than talking
Yeah.
about early life. Yeah. Any questions for Kai?
Why long ago, I think it was that most believers had such as life that they could at least have a lot of mineral that hydrogen mineral that have a lot of water. Why don't you go and get some organisms and extreme files that we might persist on Mars? Get some that are extremely that are drought tolerant and low surface. I mean, I'm not a biologist, but it seems to be. I don't understand why. The question is, do you want to have UV light?
Well, they always did at 800 as long as they have an energy source. So this question is already answered. If there's an energy source on Mars and there's liquid water and it has been light, then there's a good possibility that it's still there. But this is not we cannot take this. We can never check it. What we can do now is just we can look for the system. Then we can tell how will it disappear? If it has been there, is it still a possibility to detect it? Do these kinds of experiments. Another thing is that the low diversity cells, and there are all kinds of material present that we can use. And by putting them under, we can select for organisms which are particularly adapted to these kinds of conditions, and we can then go and isolate the organisms from the experiments and study why are these to survive these conditions, and then we can learn something about the adaptation of these organisms. We can learn about repair mechanisms, we can learn about drought resistance and so forth. And until now, what people have, there are already organisms known that would fulfill the demands that you have. There are bacillus, for example. There are organisms. They are studied. But there are several groups in Europe and also in the US that are doing these kinds of experiments. We have chosen, as one of the few groups, not to concentrate on one type of organisms, but to pick up a very complex community. And I can tell you that only less than 1%, or well, nobody really knows, but a very, very little proportion of microorganisms are known. There's a lot to learn about. So nobody really. There are numbers around, but nobody knows if they are correct. Yeah. So the only thing that is sure that we know very little about microbes when it comes to the end. So there are plenty of microorganisms out there which are very interesting in themselves to be studied, to learn more about these.
Just try to rephrase the question, but then to take organisms that might be similar to what you would expect, that some that could live in extremely, take colonies that can live under extreme conditions, drought or long ways.
Yeah, but these organisms, we picked them up in the first experiment. Most of these spore-forming organisms that we actually found have a strategy to survive a very different source, and then this is going to be
Mass soil. I mean, there's be a huge difference between that's being existed for billions of years and more highly oxidized Martian soil. It's made with soil.
But there are different types of soils that are used as analog soils. They resemble in some respect, and then there are other aspects which are different. But there's not so much known about the soil on Mars. Probably there's one of the there's also so there are different types of soil around.
Was any of these experiments with simulated Mars soil, soil in the Viking that in 76 on the server?
I think some of these experiments have actually been repeated with soils which is used for Mars analog soil, as Mars analog soils, and they get exactly the same results in organic basis. So and there's a quote on
Move the message to a practical comment before I release you for coffee break. As Axel said yesterday, he thought we should walk to the restaurant. He said, you might have your walking, you might have walking in. So, and then Anders has volunteered to take people by bus to show them where the restaurant is. Right outside the door there's a map that the secretary has prepared which shows you where the restaurant is and also gives you the bus route if you have other things you want us to do. I'll be going with Jeffrey to the Maritime Hotel by a vehicle, so if any of the other people living at the Maritime Hotel wants to come along, then meet us for the session until 6. We will rejoin here at 4:15 compared to the time I came here.